All right, thanks for joining me for how to make your yard better for birds in the environment. My name is Ashley Kennedy. I'm an entomologist, and I've included my Twitter handle here in case you want to keep in touch later. And for this presentation, I'm going to share some of the things I learned when I was a graduate student at the University of Delaware. So I'm going to start out with some kind of scary statistics. Back in November 2018, the New York Times Magazine first reported on what they called the insect apocalypse. And this was based on a meta-analysis that had just been published of 73 independent reports of insect declines around the world. And this decline in insects is actually something that people have noticed just in their own lifetimes. So if you can remember what it was like driving down the highway in the summertime 20 or 30 years ago, you'll remember that you had to clean your windshield at the end of that. And you rarely have to do that anymore. So while that might be good news for your windshield, it's actually bad news for pretty much everything else on Earth. And numerous drivers of this decline have been documented. We know that habitat loss and pesticide use and even light pollution are among them. And I'm going to include one more headline. This one's from The Onion, which, as you probably know, is a satirical source. It says, ecologists urge birds to avert global decline of insects by adopting a seed-based diet. So again, that is satire, but I think satire works best when there's an undercurrent of truth to it. And the undercurrent of truth here is that the vast majority of birds, or at least terrestrial birds, rely on insects. So even the birds that you're used to thinking of as fruit eaters or seed eaters, they have at least one important time of their life, the breeding season, when they're going to dramatically shift their diet to take advantage of all the extra fats and proteins that they can get from insects to rear their young. So going along with these insect declines, there have also been declines and mass die-offs of birds. So there was one even last December 2020. And according to this source, the mass die-off was made worse by unseasonably cold weather, and that was basically that it killed off the insects, and so these birds did not get enough nutrition that they needed during the summer to prepare for their fall migration. Here's some other headlines about bird declines. So this one famously, or it references the famous study that found that there was a decline in about 3 billion birds in North America alone in just 50 years. And then these declines are not linked just to insects and birds. So more broadly, wildlife is facing the same decline. So 1 million species face extinction, UN report says, and humans will suffer as a result. This is from the Washington Post. And when we talk about this problem, it's not just a wildlife problem. It very quickly becomes a human problem too. So here's a quote from that article. The decline in biodiversity is eroding the foundations of our economies, livelihoods, food security, health, and quality of life worldwide. All right, and so just looking at birds again, just looking at North American birds, we know that about a third of them, or more than a third of them, I should say, 432 species are at risk of extinction. And this is due to habitat loss and degradation. It's due to predation from feral and domestic cats. It's due to collisions with vehicles and buildings and towers. But I think an oft overlooked factor in this decline is a decline in that major food source that I talked about, insects. So we know that 96% of North American terrestrial birds rear their young on insects. So for my PhD project, working in Doug Tallamy's lab, I wanted to figure out which insects are the most important to breeding birds. Because if we knew that, we could create and maintain better bird habitat, give them a better shot at survival while they're facing this onslaught of all these different stressors. And the reason I say that we'd be able to maintain better bird habitat if we knew what insects they were eating, it's because many insects are host specialists and they need a particular plant lineage to survive. So I've included a picture of the famous example of the monarch butterfly on milkweed, but that example is not the exception, it's actually the rule. So Most insects are herbivorous, so feeding on plants. And then within insect herbivores, about 90% of them are host specialists. So they can't eat just any plant in the landscape. They have a particular lineage that is necessary for them to complete their life cycle. So if we could identify which insects are the most important to birds, we can go one step further in the food chain and make sure that we're planting the host plants that those insects need to survive and reproduce and support the birds, from the bottom of the ground, the bottom of the food chain on up. And I was far from being the first person to look at what types of insects birds were eating, but I wanted to briefly summarize some of the shortcomings in earlier studies. So one common method that was used in the past was neck collars or ligatures, which exactly what it sounds like. Often a zip tie or a wire would be tied around the neck of a nestling bird, and it would prevent them from swallowing their food. And then you could remove the food parcel and identify it. So as you can imagine, it's a very intrusive method, and it's difficult to apply, and it takes a lot of expertise, because if it's done wrong, it can result in the unintentional death of the bird. So I wanted to steer clear of that. There's also fecal analysis. So you can collect bird fecal matter and basically comb through it looking for the undigested remains of their prey. And this works better for some groups than others. So some insect groups are easily digested. So think of a soft, squishy caterpillar. There's not much left at the end of that process. So it would be underrepresented in the fecal matter compared to something like beetles, which have these hard outer wings that are not easily digested. So beetles tend to be overrepresented in these samples. Another method is gut dissection analysis. I wanted to avoid that for obvious reasons. I didn't want to kill these birds while they're already dealing with enough other stressors. There's also a technique called molecular scatology. So again, you're collecting fecal matter, but instead of just looking at it, you actually do genetic analyses to figure out what their prey are. And this has a lot of promise, but at the time I began this project in 2015, it wasn't yet at the place where I needed it to be, both in terms of affordability. It was not accessible for my grad student budget, and then also it just hadn't been very well developed yet. So most insects would not have been represented in the genetic database, so to speak. So instead of all those different methods, we decided to try photography. Because photography, if you get a sharp enough picture like this one here, it provides an unambiguous record of the prey. So looking at this picture, we now know that eastern bluebirds eat crab spiders. It's non-invasive. I have heard the argument that the presence of a photographer could subtly influence a bird's behavior, but compared to actually handling the birds, tying a zip tie around their necks, I felt pretty comfortable with this being minimally invasive. And it can even be inexpensive. Cameras are getting more affordable all the time. Pretty much everyone has one on their phone these days. Compared to whenever this ad came out, I'm going to guess at least a few decades ago, which one of my friends sent me. I just got a kick out of it because it made me grateful that the cameras I was using are very portable compared to this one. So for my main study species, I decided to focus on eastern bluebirds for the following reasons. They're highly insectivorous, so I knew I would get good photos of them eating insects. It's easy to find their nests because they are cavity nesters, so they will readily occupy these man-made nest boxes, of which there already were dozens or hundreds set up in my study site. Bluebirds have 2 broods a year in Delaware, so that gave me plenty of opportunity to get photos both in the spring and the summer, so a nice long field season. And then perhaps most importantly, they habituate quickly to human visitation, so I felt like I wasn't disrupting their routine too much. As you can see from this photo, I had just set up the camera just a moment before, and I was still walking away, and this female bluebird had already resumed her foraging activities. So my field site was the beautiful Mount Cuba Center in Hockessin, Delaware. I highly recommend you check it out. They have a botanic garden with an emphasis on native plants. And then in addition to the garden, they also have natural lands, which are minimally managed. They just do some invasive species removal and they mow twice a year. So I figured this was about as pristine a bluebird habitat as I was going to be able to find in the densely populated northern Delaware area. I used GoPro Hero 3 cameras, which took photos, and then I identified the prey in those photos to the lowest possible taxonomic rank. Now, if you are familiar with GoPros, you know that they're not actually trail cameras. So a real trail camera would take photos on a basis of either, they would be motion activated. And the GoPro Hero 3 is not that sophisticated, but it can be set to take photos on a time-lapse setting where it takes 1 photo per second. So as you can imagine, this generates many thousands of pictures each day, and most of them are not actually useful because they're not going to capture that critical moment when the bird has prey and is bringing it back to the nest. But even after deleting all the photos I didn't need, I had plenty of material to work with. So my first summer, I recorded 12 bluebird broods. The following year, I did 16, and then in the third and final year of the study, we did 10 more. So out of almost 10 million photos that were taken, again, most of them were not useful, but the useful photos documented more than 8,200 feeding events. So I described a feeding event as a parent bluebird returning to the nest with some kind of prey in its bill, which it then delivered into the nest. So out of those 8,200 prey, I was able to identify 93%, at least to the ordinal level. So orders are pretty broad groupings. So I could tell, for example, that prey was a moth, or it was a beetle, or a grasshopper. And in many cases, I was able to identify them below the ordinal level to family or genus or even species. So this one, for example, was sharp enough to get all the way down to species. So at a glance, this is what the eastern bluebirds were eating based on the prey that I was able to identify. And as you can see, Lepidoptera was the biggest food group. Lepidoptera is the order that includes moths and butterflies, but in their larval stage, when of course they're called caterpillars, that's when they're the most appealing to birds. So the majority of Lepidopteran prey were caterpillars. Orthoptera was the next biggest group, making up 25% of the diet. Spiders made up 19%, followed by earthworms at 5%, beetles at 3%. All of the other arthropod types combined made up 6%, and then berries and other plant material made up just under 1%. So I have some photos from that project I wanted to share, and I'm going to go through each of those prey groups in order of their representation in the diet. So beginning with Lepidoptera, which again was about 40% of the diet. So as you can see, the caterpillar diversity was pretty impressive, all sorts of caterpillars, but there was an apparent bias, at least by the numbers, towards caterpillars that are smooth and green. And just intuitively, this makes a lot of sense because smooth green caterpillars basically lack chemical and physical defenses. They don't have spines, they don't have toxins. They're really just relying on camouflage as their anti-predator defense. So if a bluebird can get a hold of one, if they spot it, it's pretty much a perfect parcel of food with nothing dangerous or anything that would make it unpalatable. Moving on to Orthoptera, this group includes the grasshoppers and crickets and katydids, which were all well represented in the diet. The biggest surprise here for me was seeing some mole crickets. So mole crickets are fossorial, which means they live underground, they dig with their front legs, and they very rarely come to the soil surface. And this was a species that I never saw when I was out in the field, but the bluebirds seemed really well adapted to home in on them when they came up to the surface because they preyed on them numerous times. The spiders, again, were 19% of the eastern bluebird breeding diet. And this included orb weavers, jumping spiders, crab spiders, and especially wolf spiders, the largest ones. The other groups in order of declining importance. Earthworms were 5% of the diet, which made it the largest non-arthropod group. Beetles made up 3% of the diet, and that included larvae and adults, both represented here in these photos. Flies, the order Diptera, made up about 1%. Almost all of that was crane flies. Berries made up less than 1%. Praying mantises made up less than 1%. However, I feel like if I could have figured out a way to measure the prey by volume, their representation in the diet would have been ranked higher, because some of the praying mantises that they brought back to the nest were enormous. The true bugs, the order Hemiptera, made up less than 1% of the diet. This included stink bugs and cicadas. I'm thinking that 2021 is going to be a good year for bluebirds and many other passerines and other wildlife in general, because of the emergence of brood 10, the periodical cicadas. Hymenoptera is the bees and wasps and ants, and they showed up occasionally, as did centipedes and millipedes. Once in a blue moon, the bluebirds ate dragonflies. This was an interesting observation because not only did they eat adult dragonflies, they were also seen occasionally with the aquatic nymphal dragonflies. And that observation had never been published before. So we actually have a little scientific note that we wrote about the first documented case of bluebirds feeding on immature dragonflies. So we know that the bluebirds don't actually forage in the water. So what must have happened for this to take place was that these dragonflies, which have a lengthy life cycle, they probably had been underwater for 3 to 5 years based on this particular species' life history. And then on the very day that they finally emerged from the water, having evaded predators in the water successfully for all that time, they came out to eclose as adults and got snatched up by a bluebird. So very unlucky for the dragonflies, but lucky for me because I got to see such a cool interaction. Isopods or woodlice showed up once in a while, as did snails and slugs. And then lastly, there were 7 instances of bluebirds feeding on vertebrates. So either frogs or, in one case, a juvenile snake. As you can see, the snake is very small. Otherwise, I don't think that would have happened. And that was also a novel observation. So you can look for it in the journal, or you can look for it in a herpetological notes. So having seen what the bluebirds were eating, it kind of raised the question, well, are they eating certain things because they like them better? Or are they just taking whatever they can get in the environment? Because Dr. Tallamy and my other professors on my graduate committee cautioned me against saying, as I often did, that the bluebirds liked caterpillars. And I would try and argue, well, of course they like caterpillars. I've got thousands of pictures of them eating caterpillars. And they reminded me, that's not good science to make that kind of assumption. So just because they're eating lots of caterpillars doesn't necessarily indicate they prefer them over other types of food. So I had to design some kind of experiment to tease out their preferences. So I dove into the literature and I found something called the Paired Stimulus Preference Assessment. It's also called a versus test because you can just think of it as food A versus food B, which one tastes better. And this test actually originated in the pet food industry, because if you think about it, it's the same kind of situation where you're trying to suss out what type of food is preferred, but you can't just ask the subject, right? You have to come up with a more creative way to measure it. So the pet food industry had already tackled this problem decades before. So there's something called first choice analysis, which is just a simple observation of which food does the animal approach first, because that tells you that regardless of how nutritious or how tasty it is, there was something about that food that was inherently more appealing to the animal. And then there's also the preference ratio analysis. So you can measure the total volume of type A that was consumed, and then compare that to the total volume of type B that was consumed. And assuming that you provide the same quantity and make it equally accessible to the bird or to the animals. So that was my goal with this study. I used 5 different types of insects, and I put them out in pairs. So in any given trial, there would be 12 insects, so 6 pairs. There'd be one type versus another type. They would be evenly sized and evenly accessible. So I used cabbage loopers, waxworms, crickets, mealworms, and stink bugs. And I chose these taxa both because they represent a broad phylogenetic sampling, and also because these are species that are available either for purchase in bulk quantities or were available for capture in large quantities. In the case of stink bugs, I used the brown marmorated stink bug, which is an invasive species in the mid-Atlantic. And when I called up my friends and asked if I could come and collect stink bugs out of their homes, they said, sure, take as many as you want. So again, I used the GoPro cameras just to record the sequence in which the birds came and selected their prey. And so they yielded photos that looked much like this one. And I'm actually skipping a whole lot of trial and error of other experimental designs I tried that just utterly failed, but this was kind of the winning strategy. So for this experiment, I recorded 23 bluebird pairs. I did 270 trials, and I found that the waxworms, which are a type of caterpillar, were the most preferred prey out of those 5 food types in a majority of bluebird subjects. And stink bugs were the least preferred in almost all of them. So from left to right, this shows their ranking from favorite to least favorite. So we've got the waxworms, which again are a caterpillar, mealworms, which are larval beetles, crickets, cabbage loopers, which is a different type of caterpillar, and brown marmorated stink bugs. And to me, the biggest surprise here was that the smooth green caterpillars, the cabbage loopers, were not more popular. And if I had to guess, and this is just speculation on my part, I was not able to further test this, but I suspect that it's because this species has a high moisture content, which in a living insect would be an asset. There's some evidence that birds do like insects with a lot of moisture, but these insects were not alive. They had been freshly killed. And so the species with more moisture decomposed very rapidly. So if I put them out on the roof of the nest box on a hot August day, then they would very quickly shrivel up. The color would change dramatically from green to black, and they would look, at least to my non-avian eye, they would look very unappetizing. So I think if I could have found some way to use even fresher specimens or some way to maintain their lifelike appearance, we might have gotten different results. But that, again, is just speculation on my part based on what I actually observed. The cabbage loopers were not preferred. So switching gears a little bit here, by this point, I knew what the bluebirds were eating and had even demonstrated that they preferred certain types of food over others. But I wanted to go beyond just bluebirds and see if these same patterns would hold over different bird species and different bird groups. So we launched this citizen science or community science project where we were basically crowdsourcing photos of anyone who had pictures of North American birds with some type of arthropod prey. And we would ask them to upload them to this website, whatdobirds.eat.com, which is no longer running. And we would ask for some basic details about when and where the photo was taken so we could add it to the official database. We also had a Facebook page. This page is still up and running. So if you happen to have any photos of birds with their insect prey, please visit it and upload them. Or if you would rather not share it publicly, you can also send the photos as a message if you click on the send message button on the right side of the page. So from that crowdsourced data set, we had 7,000 photo submissions representing 322 North American bird species. And they came from all 50 states, most Mexican states as well, most Canadian provinces. And I'm going to share some of those photos with you now. This is my favorite part because I get to show some really remarkable photos. I just wish I had more time to spend on them. Some results, just at a glance, Lepidoptera was the largest arthropod prey group for 15 of 20 examined bird families. More than 1 in 3 photos showed Lepidopteran prey. And out of those Lepidopteran prey, 85% of them were caterpillars. They were in the larval stage, not adult moths or butterflies. So I'm going to go through one bird family at a time. For each one, I start out with just a simple pie chart showing you what they were eating. And I know it's a lot of information to take in, especially because I'm going to be rolling through these slides very quickly. But what I want you to pay attention to is the bright green slice of the pie, because that tells you how important Lepidoptera was to that family. So in Corvids, based on just 26 photos, not a whole lot for this group. I'll have the N in each right corner. Based on these 26 photos, Lepidoptera made up 15% of the breeding diet. So here are some examples of that. There's a blue jay with fall webworm in Quebec, Steller's jay with a caterpillar in California, and just for something different, here's a Florida scrub jay with a centipede in Florida. Moving on to the woodpeckers, based on 145 photos that were submitted, we know that Lepidoptera made up 21% of the diet, which tied it with Hymenoptera as being one of the most important groups in their breeding diet. So here's a downy woodpecker with a Carpenter Miller caterpillar in Ontario, a downy woodpecker with a beetle larva in Alabama, and then lastly, just to mix it up, here's a Gila woodpecker with a honeybee in Arizona. Now, the family Tyrannidae is the flycatchers. So with a name like that, I thought that flies or the order Diptera would make up a big part of their diet, but that was actually just 9%. However, they did like other types of flying insects, which included moths and butterflies. So Lepidoptera was actually the biggest insect group in their diet based on almost 400 photos. So here's an eastern phoebe with a fly in Delaware, an olive-sided flycatcher with Horace's dusky wing. So that's a Lepidopteran in Iowa. And here's an eastern wood pewee with a brown marmorated stink bug. Moving on to the mockingbirds and allies, Lepidoptera was the most important arthropod group in this family as well, at 29% of the diet based on 133 photos. So here's a gray catbird with a caterpillar and a northern mockingbird with a variegated fritillary caterpillar. And one more, a long-billed thrasher with a caterpillar. Now the wrens also apparently favored Lepidoptera. They made up more than a third of the diet. This is coming from more than 400 photos. Here's a house wren with a caterpillar and a Carolina wren with a caterpillar. And then for something different, here's a Buick's wren with a katydid. Moving on to the cardinals and allies, based on 178 photos, we saw that caterpillars were the biggest arthropod group, again, 35% of the diet. So more than 1 in 3 photos showed a cardinal with a caterpillar. So there's a northern cardinal, and then here's a summer tanager with a Luna moth caterpillar. I feel like this little bird with this huge caterpillar looks like he really won the jackpot. And then lastly, here's a scarlet tanager with a bald-faced hornet. Just an interesting observation about this group is that more so than any other bird family, they did not seem deterred by insects that have sharp stingers or urticating hairs, and they often fed on wasps and other hymenopterans. The blackbirds and orioles also had Lepidoptera at 35% of their diet, making it the biggest group by far. This is based on 357 photos. So a few examples of that. Here's an eastern meadowlark with a Tursa sphinx moth caterpillar, a Baltimore oriole with a dagger moth caterpillar, and a common grackle with a whole bill full of at least 10 different fall cankerworm caterpillars. Now the thrushes, which is the family that includes bluebirds, was one of the most frequently submitted bird families. We had almost 1,000 photos coming from our community scientist team. And based on those photos, Lepidoptera made up 44% of the diet. So here's a mountain bluebird with a caterpillar, a wood thrush with a caterpillar, and then lastly, this American robin has several different prey, but including the one at the apex of the bill that's the easiest to see. It's an imperial moth caterpillar. There's also a cicada in there and a bit of an earthworm too. The warblers were represented by more than 1,000 photos. I know this family is very popular with birders, and in this family, Lepidoptera was even more important, making up more than half of the prey and submitted photos. So here is a bay-breasted warbler with a caterpillar, and a Kirtland's warbler with a caterpillar. And lastly, a prothonotary warbler with a couple of caterpillars and a couple of true bugs as well, the order Hemiptera. Now the cuckoos seem to prey on caterpillars even more. Lepidoptera made up 61% of their diet based on 66 contributed photos. There's a yellow-billed cuckoo with a Catalpa sphinx moth caterpillar. And even out in a more arid environment, where I would have expected their diet to include more orthopterans, and some insect groups flourish more in arid environments, but even out in Arizona, this greater roadrunner was feeding on a tobacco hawk moth. In a totally different setting, we have a mangrove cuckoo in Florida feeding on a golden silk orb weaver spider. Moving on to the vireos. In this family, Lepidoptera made up 62% of the breeding diet, and this is based on 162 photos. Here's a Cassin's vireo with a caterpillar, a blue-headed vireo with a southern tussock moth caterpillar, and lastly, just for something different, here's a red-eyed vireo with a spotted cucumber beetle. The New World sparrows preyed on Lepidoptera to the tune of 65% of their total breeding diet. This is based on 575 photos. So here's a dark-eyed junco with caterpillars, 3 different caterpillars at least, or possibly 4. A spotted towhee with a hornworm caterpillar. And lastly, a chipping sparrow with 3 caterpillars and a little crane fly as well. And I saved chickadees for last because this family, Parody, preyed on Lepidoptera more than any other family. 77% of their breeding diet was Lepidoptera. This is based on 238 photos. And this actually corresponds really well with previous literature, which suggested that a single pair of chickadees to rear 1 clutch of chicks would need between 6,000 and 9,000 caterpillars. Really an astonishing number. So here's a Carolina chickadee with a caterpillar. In Virginia, here's a tufted titmouse with a caterpillar. It's one of my favorite photos. And then a bridled titmouse with a cicada. All right, so having collected all of those photos from an amazing team of community scientists, it kind of raised the question of, does this method really work? How good is the data that we're getting from the public? And we thought that there would be some inherent bias in this method. So for example, maybe a photographer is only going to notice larger prey. Maybe they won't be taking pictures when the birds have particularly small prey or prey that's drab colored and harder to see. So we decided to kind of test how well this is working by taking just the photos of bluebirds, eastern bluebirds that were submitted from the public, which there were 523 of. And then we compared the breakdown of the diet in those photos with the 7,173 photos from my field project. So my project, because I was using camera traps and not an actual human photographer, we felt like this removed some of that inherent bias. And as you can see, these pie charts are eerily similar. In both data sets, Lepidoptera was the most important group at about 40% of the diet, followed by Orthoptera at about 25%, and spiders at between 15 and 20%, followed by beetles. And when I ran Oh, and followed additionally by flies at about 1% of the diet. When I actually ran statistical analyses to see if there were differences between the proportions here, there were a few small differences. So for example, beetles were better represented in the community science project than they were in my project that was restricted to just Delaware bluebirds. But I think it really speaks volumes about the value of community science that they were so similar and had the same 5 arthropod groups in the same order of prevalence in the diet. So I think this goes to show that if you get a large enough data set, it doesn't matter who collected it, these don't have to be trained scientists. They are able to gather meaningful and valuable data. And so the last part of my project, we wanted to figure out why caterpillars. Why are they so special? Why do birds like them so much? Well, the literature suggests that there are many reasons. It could be because they have low chitin in there, so their exoskeleton isn't as tough and chewy and undigestible as it is in other insects. They're high in fat and high in protein and micronutrients such as calcium and potassium and vitamin A, and they're also rich in carotenoids. And we hypothesized that birds were specifically seeking out insects with high levels of carotenoids because, well, I'm going to go through several reasons that carotenoids are important. So for one thing, they stimulate the immune system. And this is not just for birds, this is for all vertebrates. We also need carotenoids. We can't synthesize them ourselves, so we need to get them from our diet. They also improve color vision. So I know when I was a kid, my mom always told me to eat carrots to improve my vision, and it turned out she was onto something because beta carotene is found in carrots and it does improve your vision. Carotenoids can increase your sperm vitality, and they can protect your DNA from oxidative damage. And then lastly, if you're a bird, they produce red, orange, and yellow pigments in plumage, and that can be really important in helping birds to signal their quality as a prospective mate and help them to find a mate. So all else being equal, a bird that's really brightly colored with these reds and oranges and yellows, it's probably healthier than its duller counterparts because it's been getting a lot of carotenoids. So you know that it has these other benefits too, like a strong immune system. So with colleagues, I collected a whole bunch of insects. One summer, we used many different methods to try and get as much diversity as we could and as much volume as we could. And we shipped all of these insects to Arizona State University to the lab of Dr. Kevin McGraw, who's a world-class expert on carotenoids. So he ran what is called high-performance liquid chromatography analyses, which basically separate and quantify and identify the components of a mixture. So these are the results. We found that the total carotenoid content in these insect groups did vary tremendously by different taxa. So sawflies and caterpillars, which are the first 2 bars here, had much higher carotenoid content than the other groups, which included Orthoptera, the crickets and grasshoppers, the true flies, Diptera, the adult Lepidoptera, which we separated from the caterpillars, earwigs, spiders, ants and other Hymenoptera, stink bugs and other true bugs, the daddy longlegs, which are their own order, praying mantises, beetles, millipedes and centipedes, and lastly, earthworms. And when we compare the carotenoid content to what the birds are actually eating, and this is based on the many photos I have of bluebirds with their prey, we found that they do seem to prey more on insect groups with higher carotenoid content. So you can see caterpillars are both the highest in carotenoid and the best represented in the birds' diets. And then in the lower left, you can see a bunch of groups that were low in carotenoid content and not very popular with the birds, such as millipedes, and earwigs are down there too. All right, so just to briefly recap what we learned, we've learned that caterpillars are super important to birds, and we already knew going into this that they're extremely host specific. So remember, about 90% of insect herbivores, which includes caterpillars, are host specific. So what that means is our native caterpillar species need native plants to survive. So as our landscapes become invaded with these non-native invasive plants, they're no longer suitable habitat for caterpillars. They can't just suddenly switch over to a new plant that they don't share an evolutionary history with. They'll just die out. And some good news is that this is a study that came out of Dr. Tallamy's lab. This was done by Dr. Desiree Narango, as well as Dr. Tallamy and Kimberly Shropshire. They found that a relatively few genera of plants are what we now call keystone plants, or very important plants, because they support the majority of Lepidopteran species. So we don't have to worry about planting every single possible native plant that we can. We can focus our efforts on a few key genera. And I love this comic strip or infographic, for lack of a better term, that was done by Desiree Narango and Elsa Cousins, which kind of communicates the key points of that paper that I just referenced. So the majority of insect herbivores, like caterpillars, are specialized to feed on just one or a few host plants. And they found that few plants support the majority of caterpillar species. So in effect, 14% of plants are supporting 90% of the Lepidoptera. So in each county they examined, certain plant genera were consistently important for supporting caterpillar diversity, sensitive species, and food web stability. So we call these genera keystone plants. So if we include keystone plants in managed ecosystems, we will more efficiently restore insect diversity and support animals that need insects to survive. So to give you a jump start on that, I wanted to point your attention to this really cool web tool through the National Wildlife Federation. It's called the Native Plant Finder. And I have a lot of fun with this website. You just plug in your zip code and it will populate for you a list of the best plants for your area. And it will rank them in order of how many Lepidopteran species they report. So you can see the little butterfly logo with a number by it. So for my zip code, for example, goldenrod supports the most caterpillar species. And it will also generate a list of trees as well. So I'm hoping that as a society, we can shift away from landscapes that look like this one, which is admittedly tidy. But when I look at this picture, I just think of it as a food desert. Imagine being a Carolina chickadee and you've got to find 6,000 to 9,000 caterpillars to rear your babies, and it's just not going to happen in a landscape that looks like this. But it could happen in a landscape that looks more like this one, which is, to my eye, much more interesting and much more beautiful. And with that, I'm just going to leave you with this take-home message: to conserve birds, we need to plant a wide variety of native plants. I want you to garden as if life depended on it, because it does. And with that, I'll just leave this slide up for a moment to acknowledge everyone who helped me with this project, which included my graduate committee at the University of Delaware, Mount Cuba, which was my beautiful field site, Daniel Hildreth, who generously funded this entire project, and numerous additional colleagues who provided support in various ways. And then last, but certainly not least, more than 1,200 community scientists who contributed those beautiful photos that I showed earlier. So I like to tell people I probably had more help from more people on my project than anyone else who's earned a PhD, and I am immensely grateful. So with that, I'd be happy to take any questions, and I wanted to thank you very much for your time and attention.